Alright guys, here is the summary that you asked for over photosynthesis. Try and keep this as short and sweet as possible. Okay, so let's start with the light-dependent reactions. So pretty much this part is just electron transport chain. What you have to realize is that there are two ways that electron transport chain can happen. It all starts the same way. We're going to utilize a water molecule. That water molecule is going to get split. It's going to be split into its hydrogens and into its oxygens. The oxygen is going to leave via the stomata and becomes atmospheric oxygen that we're going to be able to breathe in at some point. Now the hydrogens are going to go on a bit of a journey. The hydrogens have electrons on them. Those electrons are needed to start off the electron transport chain. So the hydrogens get stripped of their electrons. The hydrogens are pumped into the thylakoid space where we can create that hydrogen gradient that the electron transport chain is known for, and those electrons get sent into the various reaction centers of the photosystem so that they can be excited. Now, once we get to that point, that's where we can have some differences. We can either go through non-cyclic electron flow, or we can go through cyclic electron flow. So let's talk about what the differences are. Non-cyclic electron flow is in and out. So that's the one that's going to utilize, I'm going to try and write on this, oh Lord. That's the one that's going to utilize two photosystems. So we're using both photosystem 1 and photosystem 2. Then we are going to, like I said, we're going to split water through photolysis, and that splitting of the water is what replenishes or replaces those electrons in those photosystems each time. So H2O is split. Okay. Once we split H2O, we're going to make oxygen. So O2 gets made. At the very end of the process, we're going to make NAD, whoops, that should be a D, DPH, which is our electron carrier. And then as we're passing those electrons through, as those electrons are moving and being attracted to those proteins that are electronegative, we are going to be creating ATP. That all happens in non-cyclic electron flow. Now in cyclic electron flow, we only use one photosystem. And in this one, we're going to use photosystem 1. So PS1 is the photosystem we're using. The water is not going to get split every time. Water only gets split once in this system, in cyclic. It only happens one time. So electrons are only replenished one time. So water is not split to replace electrons. Okay, we're not splitting water a second time, which means that oxygen is not going to be released. If we don't split oxygen, oxygen not released. Now, the very final product of the electron transport chain, if it happens in one motion, is to make NADPH. We are passing those hydrogens on to NADPH. This is not going to happen. So NADPH is not made, not produced. Okay? The one thing that will be made is going to be ATP, because we're just going to keep cycling those electrons back to photosystem 1 every single time and having them run down the electron transport chain, continuously making ATP. All right, don't forget that the whole point of going through the light-dependent reactions is so you can make your NADPH, your electron carrier, and your ATP, your energy. We're going to need both of these in order to make glucose, which is going to happen in the Calvin cycle. So let me see if I can find a way to add. I think I added a page, hopefully. And now let's talk about the Calvin cycle. Whoops, Trying to write with my laser. Okay, so in the Calvin cycle, it's a 3-step process. Remember, it turns 3 separate times, 1 for each molecule of carbon dioxide. So happens, 3 times. Okay, so we're going to do this as a summary, so I'm going to talk about it like everything's happened. We've done all 3 carbon dioxides and everything. So we require 3 CO2 molecules. That's coming from the atmosphere. Okay, those 3 carbon molecules are going to be added. So these are added to RUBP. And RUBP has 5 carbons. This is done by the enzyme Rubisco in C3 plants. If we're talking about C4 plants, then we're talking about the enzyme pepcarboxylase. Okay, so this is carbon fixation. We are taking the carbons off of those carbon dioxide molecules and adding them to a 5-carbon molecule to make a 6-carbon molecule. I'm pausing like you can answer me, but anyway. All right, now don't forget, 3 separate times, 1 for each carbon dioxide molecule that we take in from our atmosphere. So that's step 1. We add another step. Step 2, so that step 1 was carbon fixation. Step 2 is reduction. So we've just made 3 6-carbon molecules. These molecules are very unstable. So they're going to split in half, and that's going to give us 6 3-carbon molecules. Okay? Now, remember we made NADPH when we went through light-dependent reactions. So our NADPH is going to reduce our 6 3-carbon molecules. Okay? So let's try that again. We make 3 6-carbon molecules when we do carbon fixation. Those molecules are large and they're unstable, so they will tend to break apart. When they break apart, we end up with 3 carbon molecules, but we have 6 of them. NADPH that we made in the light-dependent reaction is now going to reduce those 6 3-carbon molecules, so we're going to add some hydrogens to it, and this is going to require some ATP. So we need to reduce them, and we have to rearrange them, so we need ATP. Once rearrangement has taken place, we have now made 6 3-carbon molecules, which are known as G3P. G3P is going to be the precursor for glucose. We're going to use that to make glucose. Now, once your G3P is made, it is going to leave the cell so that we can use it to make glucose. Now we need to do step 3. Step 3 is the regeneration of RUBP. Okay, we need to rearrange those carbons back into the original arrangement, the original formation, so that we can utilize the cycle again if need be. To rearrange those carbons, we need ATP. So we use ATP to rearrange remaining carbons into RUBP. So let's go ahead and recap using our photosynthesis equation. Photosynthesis equation says that 6 H2Os plus 6 CO2s will produce, using light, 6 oxygen molecules plus 1 molecule of glucose, C6H12O6. So let's talk about what all of these things are used for and where they're coming from. Oops. Our water gets split. The hydrogens are used to replace electrons. Okay, so we use the hydrogens to replace the electrons that we lose from photosystem 2. Okay, carbon dioxide is used in the Calvin cycle. And it provides carbon, okay, so that we can make organic compounds. Okay, our light is what is going to excite our electrons during our light reaction. Oxygen is a byproduct of splitting water. Sorry for the shorthand, but I'm using my old pen because I'm still at school, so it's a little bit messy. Glucose is the last thing that we're going to make, and it's the organic molecule, organic compound, ultimately made, that should be ultimately, made by the Calvin cycle. So in terms of numbers, this one is not as numbers detailed as respiration, but pretty much this is your skeleton for photosynthesis. This is the stuff you really need to know, where things came from, what they're used for. I really hope that you're studying for this test, because it is easy if you know what you're doing. But if you've been utilizing other people's help in order to do your homework, and you haven't been actively studying, it's not going to be that easy. Alright, with that being said, study hard. See y'all tomorrow.